Chapter 18 The Cell Division Cycle. Where a cell arises, there must be a previous cell, just as animals can only arise from animals and plants from plants. This cell doctrine, proposed by the German pathologist Rudolf Virchow in 1858, carried with it a profound message for the continuity of life. Cells are generated from cells, and the only way to make more cells is by the division of those that already exist. All living organisms, from a unicellular bacterium to a multicellular mammal, are thought to be products of repeated rounds of cell growth and division, extending back in time to the beginnings of life more than 3 billion years ago. A cell reproduces by carrying out an orderly sequence of events in which it duplicates its contents and then divides in two. This cycle of duplication and division, known as the cell cycle, is the essential mechanism by which all living things reproduce. The details of the cell cycle vary from organism to organism and at different times in an individual organism's life. In unicellular organisms, such as bacteria and yeasts, each cell division produces a complete new organism, whereas many rounds of cell division are required to make a new multicellular organism from a fertilized egg. Certain features of the cell cycle, however, are universal, as they allow every cell to perform its most fundamental task, to copy and pass on its genetic information to the next generation of cells. To produce two genetically identical daughter cells, the DNA in each chromosome must be faithfully replicated, and the replicated chromosomes must then be accurately distributed or segregated into the two daughter cells, so that each cell receives a complete copy of the entire genome. Figure 18.1. Most cells also duplicate their own macromolecules and organelles, and they double in size before they divide. Otherwise, each time they divided, they would get smaller and smaller. Thus, to maintain their size, dividing cells must coordinate their growth with their division. To explain how cells reproduce, we therefore have to consider three major questions. 1. How do cells duplicate their contents? 2. How do they partition the duplicated contents and split in two? 3. How do they coordinate all the machinery that is required for these two processes? The first question is considered elsewhere in this book. In Chapter 6, we discuss how DNA is replicated, and in Chapters 7, 11, 15, and 17, we describe how the eukaryotic cell manufactures other components, such as proteins, membranes, organelles, and cytoskeletal filaments. In this chapter, we tackle the second and third questions: how a eukaryotic cell segregates its duplicated contents to produce two daughter cells, and how it coordinates the various steps of this reproductive cycle. We begin with an overview of the events that take place during the cell cycle. We then describe the complex system of regulatory proteins called the cell cycle control system, which orders and coordinates these events to ensure that they occur in the correct sequence. We next discuss in detail the major stages of the cell cycle, in which the chromosomes are duplicated and then segregated into the two daughter cells. At the end of this chapter, we consider how an animal regulates the size and number of its cells, and thereby the size of the organism and its organs. We describe how animals eliminate unwanted cells by a form of programmed cell death called apoptosis, and we then discuss how they use extracellular signals to control cell survival, cell growth, and cell division. Overview of the cell cycle. The most basic function of the cell cycle is to duplicate accurately the vast amount of DNA in the chromosomes and then distribute the copies into genetically identical daughter cells. The duration of the cell cycle varies greatly from one cell type to another. A single-celled yeast can divide every 2 hours or so in ideal conditions, whereas a mammalian liver cell divides on average less than once a year. Table 18.1. We briefly describe here the sequence of events that occur in a fairly rapidly dividing, proliferating mammalian cells. We then introduce the cell cycle control system that ensures that the various events of the cycle take place in the correct sequence and at the correct time. The eukaryotic cell cycle is divided into 4 phases. Seen under a microscope, the most dramatic events in the cycle are when the nucleus divides, a process called mitosis, and when the cell later splits in two, a process called cytokinesis. These two processes together constitute the M phase of the cell cycle. In a typical mammalian cell, the whole of M phase takes about an hour, which is only a small fraction of the total cell cycle time. The period between one M phase and the next is called interphase. Under the microscope, it appears deceptively as an uneventful interlude during which the cell simply increases in size. Interphase, however, is a very busy time for the cell, and it encompasses the remaining 3 phases of the cycle. During S phase, S equals synthesis, the cell replicates its nuclear DNA, an essential prerequisite for cell division. S phase is flanked by 2 phases in which the cell continues to grow. The G1 phase, G equals gap, is the interval between the completion of M phase and the beginning of S phase. The G2 phase is the interval between the end of S phase and the beginning of M phase. Figure 18.2. During these gap phases, the cell monitors the internal and external environment to ensure that conditions are suitable and its preparations are complete before it commits itself to the major upheavals of S phase and mitosis. At particular points in G1 and G2, the cell decides whether to proceed to the next phase or pause to allow more time to prepare. During all of interphase, a cell generally continues to transcribe genes, synthesize proteins, and grow in mass. Together, G1 and G2 phases provide additional time for the cell to grow and duplicate its cytoplasmic organelles. If interphase lasted only long enough for DNA replication, the cell would not have time to double its mass before it divided, and would consequently shrink with each division. Indeed, in some special circumstances that is just what happens. In some animal embryos, for example, the first cell divisions after fertilization, called cleavage divisions, serve to subdivide a giant egg cell into many smaller cells as quickly as possible. In these embryonic cell cycles, the G1 and G2 phases are drastically shortened, and the cells do not grow before they divide. Following DNA replication in S phase, the 2 copies of each chromosome remain tightly bound together. The first visible sign that a cell is about to enter M phase is the progressive condensation of its chromosomes. As condensation proceeds, the replicated chromosomes first become visible in the light microscope as long threads, which gradually get shorter and thicker. This condensation makes the chromosomes less likely to get entangled, so that they are easier to segregate to the 2 forming daughter cells during mitosis. A cell cycle control system triggers the major processes of the cell cycle. To ensure that they replicate all of their DNA and organelles and divide in an orderly manner, eukaryotic cells possess a complex network of regulatory proteins known as the cell cycle control system. This system guarantees that the events of the cell cycle, DNA replication, mitosis, and so on, occur in a set sequence, and that each process has been completed before the next one begins. To accomplish this, the control system is itself regulated at certain critical points of the cycle by feedback from the process being performed. Without such feedback, an interruption or a delay in any of the processes could be disastrous. All of the nuclear DNA, for example, must be replicated before the nucleus begins to divide, which means that a complete S phase must precede M phase. If DNA synthesis is slowed down or stalled, mitosis and cell division must also be delayed. Similarly, if DNA is damaged, the cycle must arrest in G1S or G2 so that the cell can repair the damage, either before DNA replication is started or completed or before the cell enters M phase. The cell cycle control system achieves all of this by means of molecular brakes that can stop the cycle at various checkpoints. In this way, the control system does not trigger the next step in the cycle unless the cell is properly prepared. 3 checkpoints that control progression through the cell cycle are illustrated in Figure 18.3. One checkpoint operates in G1 and allows the cell to confirm that the environment is favorable for cell proliferation before committing to S phase. Cell proliferation in animals requires both sufficient nutrients and specific signal molecules in the extracellular environment. If extracellular conditions are unfavorable, cells can delay progress through G1 and may even enter a specialized resting state known as G0. In many cells, including nerve cells and skeletal muscle cells, remain in G0 for the lifetime of the organism. Another checkpoint operates in G2 and ensures that cells do not enter mitosis until damaged DNA has been repaired and DNA replication is complete. A third checkpoint operates during mitosis and ensures that the replicated chromosomes are properly attached to a cytoskeletal machine called the mitotic spindle, before the spindle pulls the chromosomes apart and distributes them into the 2 daughter cells. The checkpoint in G1 is especially important as a point in the cell cycle where the control system can be regulated by signals from other cells. In a multicellular animal, the control system is highly responsive to signals from other cells that stimulate cell division when more cells are needed and block it when they are not. The control system therefore plays a central part in the regulation of cell numbers in the tissues of the body. If the system malfunctions such that cell division is excessive, cancer can result. We will see later how extracellular signals influence the decisions made at this checkpoint. Cell cycle control is similar in all eukaryotes. Some features of the cell cycle, including the time required to complete certain events, vary greatly from one cell type to another, even within the same organism. The basic organization of the cycle, however, is essentially the same in all eukaryotic cells, and all eukaryotes appear to use similar machinery and control mechanisms to drive and regulate cell cycle events. The proteins of the cell cycle control system first appeared more than 1 billion years ago, and they have been so well conserved over the course of evolution that many of them function perfectly when transferred from a human cell to a yeast. How we know, pages 15 to 16. Because of this similarity, biologists can study the cell cycle and its regulation in a variety of organisms and use the findings from all of them to assemble a unified picture of how eukaryotic cells divide. Many discoveries about the cell cycle have come from the systematic search for mutations and inactive essential components of the cell cycle control system in yeasts. Studies of cultured mammalian cells in animal embryos have also been useful for examining the molecular mechanisms governing the control of cell proliferation in multicellular organisms. The cell cycle control system. 2 types of machinery are involved in cell division. One manufactures the new components of the growing cell, and another hauls the components into their correct places and partitions them appropriately when the cell divides in two. The cell cycle control system switches all this machinery on and off at the correct times and thereby coordinates the various steps of the cycle. The core of the cell cycle control system is a series of biochemical switches that operate in a defined sequence and orchestrate the main events of the cycle, including DNA replication and segregation of the duplicated chromosomes. In this section, we review the protein components of the control system and discuss how they work together to trigger the different phases of the cycle. The cell cycle control system depends on cyclically activated protein kinases called CDKs. The cell cycle control system governs the cell cycle machinery by cyclically activating and then inactivating the key proteins and protein complexes that initiate or regulate DNA replication, mitosis, and cytokinesis. As discussed in Chapter 4, phosphorylation followed by dephosphorylation is one of the most common ways by which cells switch the activity of a protein on and off. See Figure 438, and the cell cycle control system uses this mechanism repeatedly. The phosphorylation reactions that control the cell cycle are carried out by a specific set of protein kinases, while dephosphorylation is performed by a set of protein phosphatases. The protein kinases at the core of the cell cycle control system are present in proliferating cells throughout the cell cycle. They are activated, however, only at appropriate times in the cycle, after which they quickly become deactivated again. Thus, the activity of each of these kinases rises and falls in a cyclical fashion. Some of these protein kinases, for example, become active toward the end of G1 phase and are responsible for driving the cell into S phase. Another kinase becomes active just before M phase and is responsible for driving the cell into mitosis. Switching these kinases on and off at the appropriate times is partly the responsibility of another set of proteins in the control system, the cyclins. Cyclins have no enzymatic activity themselves, but they have to bind to the cell cycle kinases before the kinases can become enzymatically active. The kinases of the cell cycle control system are therefore known as cyclin-dependent protein kinases, or CDKs. Figure 18.4. Cyclins are so named because, unlike the CDKs, their concentrations vary in a cyclical fashion during the cell cycle. The cyclical changes in cyclin concentrations help drive the cyclic assembly and activation of the cyclic CDK complexes. Activation of these complexes in turn triggers various cell cycle events, such as entry into S phase or M phase. Figure 18.5. We discuss how the CDKs and cyclins were discovered in the How We Know, pages 615 to 616. The activity of CDKs is also regulated by phosphorylation and dephosphorylation. The rise and fall of cyclin levels plays an important part in regulating CDK activity during the cell cycle, but there is more to the story. Cyclin concentrations increase gradually, but the activity of the associated cyclin CDK complexes tends to switch on abruptly at the appropriate time in the cell cycle. See Figure 18.5. So what triggers the abrupt activation of these complexes? For a cyclin CDK to be maximally active, the CDK has to be phosphorylated at one site by a specific protein kinase and dephosphorylated at other sites by a specific protein phosphatase. Figure 18.9. We discuss later how these kinases and phosphatases regulate the activity of specific cyclin CDKs and thus control progression through the cell cycle. Different cyclin CDK complexes trigger different steps in the cell cycle. There are several types of cyclins, and in most eukaryotes, several types of CDKs involved in cell cycle control. Different cyclin CDK complexes trigger different steps of the cell cycle. The cyclin that acts in G2 to trigger entry into M phase is called M cyclin, and the active complex it forms with its CDK is called MCDK. Distinct cyclins called S cyclins and G1 S cyclins bind to a distinct CDK protein late in G1 to form SCDK and G1 SCDK respectively, and trigger S phase. The action of SCDK and MCDK are shown in figure 1810. Other cyclins called G1 cyclins act earlier in G1 and bind to other CDK proteins to form G1 CDKs, which help drive the cell through G1 toward S phase. We will see later that the formation of these G1 CDKs in animal cells usually depends on extracellular signal molecules that stimulate cells to divide. The names of the individual cyclins and their CDKs are listed in Table 18.2. As previously explained, the different CDKs also have to be phosphorylated and dephosphorylated in order to act. See figure 18.9. Each of these activated cyclin CDK complexes in turn phosphorylates a different set of target proteins in the cell. As a result, each type of complex triggers a different transition step in the cycle. MCDK, for example, phosphorylates key proteins that cause the chromosomes to condense, the nuclear envelope to break down, and the microtubules of the cytoskeleton to reorganize to form the mitotic spindle. These events herald the entry into mitosis, as we discuss later. The cell cycle control system also depends on cyclical proteolysis. The concentration of each type of cyclin rises gradually but then falls sharply at a specific time in the cell cycle. See figure 1810. This abrupt fall results from the targeted degradation of the cyclin protein. Specific enzyme complexes add ubiquitin chains to the appropriate cyclin, which is then directed to the proteasome for destruction. Figure 1811. This rapid elimination of the cyclin returns the CDK to its inactive state. Although the activation of CDKs triggers some of the transitions from one part of the cell cycle to the next, their inactivation triggers others. For example, inactivation of MCDK, which is triggered by the destruction of M cyclin, leads to the molecular events that take the cell out of mitosis. Proteins that inhibit CDKs can arrest the cell cycle at specific checkpoints. We have seen that the cell cycle control system triggers the events of the cell cycle in a specific order. It triggers mitosis, for example, only after all the DNA has been replicated, and it permits the cell to divide into two only after mitosis has been completed. If one of the steps is delayed, the control system delays the activation of the next step so that the normal sequence is maintained. This self-regulating property of the control system ensures, for example, that if DNA synthesis is halted for some reason during S phase, the cell will not proceed into M phase with its DNA only partly replicated. As mentioned earlier, the molecular system accomplishes this feat largely through the action of molecular brakes that can stop the cell cycle at specific checkpoints, allowing the cell to monitor its internal state and its environment before continuing through the cycle. See figure 18.3. Some of these molecular brakes rely on CDK inhibitor proteins that block the assembly or activity of one or more cyclin CDK complexes. Certain CDK inhibitor proteins, for example, help maintain CDKs in an inactive state during the G1 phase of the cycle, thus delaying progression into S phase. Pausing at this checkpoint gives the cell more time to grow, or allows it to wait until extracellular conditions are favorable for division. As a general rule, Mammalian cells will multiply only if they are stimulated to do so by extracellular signals called mitogens produced by other cells. If deprived of such signals, the cell cycle arrests at a G1 checkpoint. See Figure 18.3. And if the cell is deprived for long enough, it will withdraw from the cell cycle and enter the non-proliferating state, G0, in which the cell can remain for days or weeks, or even for the lifetime of the organism. Figure 18.12. Most of the diversity in cell division rates in the adult body lies in the variation in the time that cells spend in G0 or in G1. Some cell types, such as liver cells, normally divide only once every year or two, whereas certain epithelial cells in the gut divide more than twice a day to renew the lining of the gut continually. Many of our cells fall somewhere in between. They can divide if the need arises, but normally do so infrequently. Escape from the G1 checkpoint or from G0 requires the accumulation of G1 cyclins, and mitogens function by stimulating this accumulation. Once past the G1 checkpoint, a cell usually proceeds all the way through the rest of the cell cycle quickly, typically within 12 to 24 hours in mammals. The G1 checkpoint is therefore sometimes called start, because passing it represents a commitment to complete a full division cycle, although a better name might be stop. See Figure 18.12. Some of the main checkpoints in the cell cycle are summarized in Figure 18.13. The most radical decision that the cell cycle control system can make is to withdraw the cell from the cell cycle permanently. This is different from withdrawing from the cell cycle temporarily, to wait for more favorable conditions, and it has a special importance in multicellular organisms. In the human body, for example, nerve cells and skeletal muscle cells permanently stop dividing when they differentiate. They enter an irreversible G0 state, in which the cell cycle control system is largely dismantled. Many of the CDKs and cyclins disappear, and the cyclin-CDK complexes that are still present are inhibited by CDK inhibitor proteins. We now turn to S phase, in which cells replicate their DNA and begin to prepare their chromosomes for segregation. S phase. Before a cell divides, it must duplicate its DNA. As we discuss in Chapter 6, this replication must occur with extreme accuracy to minimize the risk of mutations in the next cell generation. Of equal importance, every nucleotide in a genome must be copied once, and only once, to prevent the damaging effects of gene amplification. In this section, we consider the elegant mechanisms by which the cell cycle control system initiates the replication process and at the same time prevents replication from happening more than once per cell cycle. SCDK initiates DNA replication and helps block re-replication. As we discuss in Chapter 6, DNA replication begins at origins of replication, nucleotide sequences that are scattered along each chromosome. These sequences recruit specific proteins that control the initiation and completion of DNA replication. One multi-protein complex, the origin recognition complex, ORC, remains bound to origins of replication throughout the cell cycle, where it serves as a sort of landing pad for additional regulatory proteins that bind before the start of S phase. One of these regulatory proteins, called CDC6, is present at low levels during most of the cell cycle, but its concentration increases transiently in early G1. When CDC6 binds to ORCs in G1, it promotes the binding of additional proteins to form a pre-replicative complex. Once the pre-replicative complex has been assembled, the replication origin is ready to fire. The activation of SCDK in late G1 then pulls the trigger, initiating DNA replication. As shown in Figure 18.14, SCDK does not only initiate origin firing, it also helps prevent re-replication of the DNA. Activated SCDK helps phosphorylate CDC6, causing it and the other proteins in the pre-replicative complex to dissociate from the ORC after an origin has fired. This disassembly prevents replication from occurring again at the same origin. In addition to promoting dissociation, phosphorylation of CDC6 by SCDK and by MCDK, which becomes active at the start of M phase, marks it for degradation, ensuring that DNA replication is not reinitiated later in the same cell cycle. Cohesins help hold the sister chromatids of each replicated chromosome together. After the chromosomes have been duplicated in S phase, the two copies of each replicated chromosome remain tightly bound together as identical sister chromatids. The sister chromatids are held together by protein complexes called cohesins, which assemble along the length of each sister chromatid as the DNA is replicated in S phase. The cohesins form protein rings that surround the two sister chromatids, keeping them united. Figure 1815. This cohesion between sister chromatids is crucial for proper chromosome segregation, and it is broken completely only in late mitosis to allow the sister chromatids to be pulled apart by the mitotic spindle. Defects in sister chromatid cohesion in yeast mutants, for example, lead to major errors in chromosome segregation. DNA damage checkpoints help prevent the replication of damaged DNA. The cell cycle control system uses several distinct checkpoint mechanisms to halt progress through the cell cycle if DNA is damaged. DNA damage checkpoints in G1 and S phase prevent the cell from starting or completing S phase and replicating damaged DNA. Another checkpoint operates in G2 to prevent the cell from entering M phase with damaged or incompletely replicated DNA. See Figure 1813. The G1 checkpoint mechanism is especially well understood. DNA damage causes an increase in both the concentration and activity of a protein called p53, which is a transcription regulator that activates the transcription of a gene encoding a CDK inhibitor protein called p21. The p21 protein binds to G1 SCDK and SCDK, preventing them from driving the cell into S phase. Figure 1816. The arrest of the cell cycle in G1 gives the cell time to repair the damaged DNA before replicating it. If the DNA damage is too severe to be repaired, p53 can induce the cell to kill itself by undergoing apoptosis. If p53 is missing or defective, the unrestrained replication of damaged DNA leads to a high rate of mutation and the production of cells that tend to become cancerous. In fact, mutations in the p53 gene are found in about half of all human cancers. Once DNA replication has begun, another type of checkpoint mechanism operates to prevent a cell entering M phase with damaged or incompletely replicated DNA, as we saw in Figure 18.9. The activity of cyclin CDK complexes is inhibited by phosphorylation at particular sites. For the cell to progress into mitosis, MCDK has to be activated by the removal of these inhibitory phosphatases by a specific protein phosphatase. When DNA is damaged or incompletely replicated, this activating protein phosphatase is itself inhibited, so the inhibitory phosphatases are not removed from MCDK. As a result, MCDK remains inactive, and M phase cannot be initiated until DNA replication is complete and any DNA damage is repaired. Once a cell has passed through these checkpoints and has successfully replicated its DNA in S phase and progressed through G2, it is ready to enter M phase, in which it divides its nucleus, the process of mitosis, and then its cytoplasm, the process of cytokinesis. See Figure 18.2. In the next 3 sections, we focus on M phase. We first present a brief overview of M phase as a whole, and then discuss in more detail the events that occur during mitosis, and then those that occur during cytokinesis. Our focus will be mainly on animal cells. M phase. Although M phase, mitosis plus cytokinesis, occurs over a relatively short amount of time, about 1 hour in a mammalian cell that divides once a day, or even once a year, it is by far the most dramatic phase of the cell cycle. During this brief period, the cell reorganizes virtually all of its components and distributes them equally into the 2 daughter cells. The earlier phases of the cell cycle, in effect, serve to set the stage for the drama of M phase. The central problem for a cell in M phase is to accurately segregate its chromosomes, which were replicated in the preceding S phase, so that each new daughter cell receives an identical copy of the genome. With minor variations, all eukaryotes solve this problem in a similar way. They assemble 2 specialized cytoskeletal machines, one that pulls the duplicated chromosome sets apart during mitosis, and another that divides the cytoplasm into 2 halves, cytokinesis. We begin our discussion of M phase with an overview in which we consider how the cell sets the process of M phase in motion. We then address mitosis and cytokinesis in turn. CDK drives entry into M phase and mitosis. One of the most remarkable features of cell cycle control is that a single protein complex, CDK, brings about all of the diverse and intricate rearrangements that occur in the early stages of mitosis. CDK triggers the condensation of the replicated chromosomes into compact, rod-like structures, readying them for segregation. And it also induces the assembly of the mitotic spindle that will separate the condensed chromosomes and segregate them into the 2 daughter cells. As discussed earlier, CDK activation begins with the accumulation of cyclin. See Figure 1810. Synthesis of cyclin starts immediately after S phase. Its concentration then rises gradually and helps time the onset of M phase. The increase in cyclin protein leads to a corresponding accumulation of CDK complexes, but those complexes, when they first form, are inactive. The sudden activation of the CDK stockpile at the end of G2 is triggered by the activation of a protein phosphatase, CDC25, that removes the inhibitory phosphatases holding CDK activity in check. Figure 1817. Once activated, each CDK complex can indirectly activate more CDK by phosphorylating and activating more CDC25, as illustrated in Figure 1818. In addition, activated CDK also inhibits the inhibitory kinase Wee1. See Figure 1817. Further promoting the activation of CDK. The overall consequence is that once the activation of CDK begins, there is an explosive increase in CDK activity that drives the cell abruptly from G2 into M phase. Condensins help configure duplicated chromosomes for separation. When the cell is about to enter M phase, the replicated chromosomes condense, becoming visible as thread-like structures. Protein complexes called condensins help carry out this chromosome condensation. The CDK that initiates entry into M phase triggers the assembly of condensin complexes onto DNA by phosphorylating some of the condensin subunits. Condensation makes the mitotic chromosomes more compact, reducing them to small physical packets that can be more easily segregated within the crowded confines of the dividing cell. Condensins are structurally related to cohesins, the proteins that hold sister chromatids together. See Figure 1815. Both cohesins and condensins form ring structures, and together The two types of protein rings help to configure the replicated chromosomes for mitosis. Cohesins assemble on the DNA as it replicates in S phase and tie together two parallel DNA molecules, the identical sister chromatids. Condensins, by contrast, assemble on each individual chromatid at the start of M phase and coil up the DNA to help each chromatid condense. Figure 1819. The cytoskeleton carries out both mitosis and cytokinesis. After the replicated chromosomes have condensed, two complex cytoskeletal machines assemble in sequence to carry out the two mechanical processes that occur in M phase. The mitotic spindle carries out nuclear division, mitosis, and in animal cells and many unicellular eukaryotes, the contractile ring carries out cytoplasmic division, cytokinesis. Figure 1820. Both structures rapidly disassemble after they have performed their tasks. The mitotic spindle is composed of microtubules and the various proteins that interact with them, including microtubule-associated motor proteins, discussed in Chapter 17. In all eukaryotic cells, the mitotic spindle is responsible for separating the replicated chromosomes and allocating one copy of each chromosome to each daughter cell. The contractile ring consists of actin filaments and myosin filaments arranged in a ring around the equator of the cell, introduced in Chapter 17. It starts to assemble just beneath the plasma membrane toward the end of mitosis. As the ring contracts, it pulls the membrane inward, thereby dividing the cell in two. See Figure 1820. We discussed later how plant cells, which have a cell wall to contend with, divide their cytoplasm by a very different mechanism. M phase is conventionally divided into 6 stages. Although M phase proceeds as a continuous sequence of events, it is traditionally divided into 6 stages. The first stage of M phase, prophase, prometaphase, metaphase, anaphase, and the first 5 stages of M phase, prophase, prometaphase, metaphase, anaphase, and telophase, constitute mitosis, which was originally defined as the period in which the chromosomes are visible because they have become condensed. Cytokinesis constitutes the 6th stage, and it overlaps in time with the end of mitosis. The 6 stages of M phase are summarized in panel 18.1, pages 626 to 627. Together, they form a dynamic sequence in which many independent cycles, involving the chromosomes, cytoskeleton, and centrosomes, are coordinated to produce 2 genetically identical daughter cells. The 5 stages of mitosis occur in strict sequential order, whereas cytokinesis begins in anaphase and continues through telophase. During prophase, the replicated chromosomes condense and the mitotic spindle begins to assemble outside the nucleus. During prometaphase, the nuclear envelope breaks down, allowing the spindle microtubules to bind to the chromosomes. During metaphase, the mitotic spindle gathers all of the chromosomes to the center, equator of the spindle. During anaphase, the 2 sister chromatids in each replicated chromosome synchronously split apart, and the spindle draws them to opposite poles of the cell. During telophase, a nuclear envelope reassembles around each of the 2 sets of separated chromosomes to form 2 nuclei. Movie 18.3 and Movie 18.4. Cytokinesis is complete by the end of telophase, when the nucleus and cytoplasm of each of the daughter cells return to interphase, signaling the end of M phase. Mitosis. Before nuclear division or mitosis begins, each chromosome has been replicated and consists of 2 identical sister chromatids, held together along their length by cohesin proteins. See Figure 1815. During mitosis, the cohesin proteins are cleaved, the sister chromatids split apart, and the resulting daughter chromosomes are pulled to opposite poles of the cell by the mitotic spindle. Figure 1821. In this section, we examine how the mitotic spindle assembles and functions. We discuss how the dynamic instability of microtubules and the activity of microtubule-associated motor proteins contribute to both the assembly of the spindle and its ability to segregate the sister chromatids. Finally, we review the checkpoint mechanism that operates in mitosis to ensure the synchronous separation of the sister chromatids, the proper segregation of the two chromosomes sets the two daughter cells, and the orderly and timely exit from mitosis. Centrosomes duplicate to help form the two poles of the mitotic spindle. Before M phase begins, two critical events must be completed. DNA must be fully replicated, and in animal cells, the centrosome must be duplicated. The centrosome is the principal microtubule organizing center in animal cells. It duplicates so that it can help from the two poles of the mitotic spindle and so that each daughter cell can receive its own centrosome. Centrosome duplication begins at the start of S phase and is triggered by the same CDKs, G1 SCDK and SCDK, that trigger DNA replication. Initially, when the centrosome duplicates, both copies remain together as a simple complex on one side of the nucleus. As mitosis begins, however, the two centrosomes separate, and each nucleates a radial array of microtubules called an aster. The two asters move to opposite sides of the nucleus to form the two poles of the mitotic spindle. Figure 1822. The process of centrosome duplication and separation is known as the centrosome cycle. The mitotic spindle starts to assemble in prophase. The mitotic spindle begins to form in prophase. This assembly of the highly dynamic spindle depends on the remarkable properties of microtubules. As discussed in Chapter 17, microtubules continuously polymerize and depolymerize by the addition and loss of their tubulin subunits, and individual filaments alternate between growing and shrinking, a process called dynamic instability. See Figure 1711. At the start of mitosis, the dynamic instability of microtubules increases, in part because MCDK phosphorylates microtubule-associated proteins that influence the stability of microtubule filaments. As a result, during prophase, rapidly growing and shrinking microtubules extend in all directions from the two centrosomes, exploring the interior of the cell. Some of the microtubules growing from one centrosome interact with the microtubules from the other centrosome. This interaction stabilizes the microtubules, preventing them from depolymerizing, and it joins the two sets of microtubules together to form the basic framework of the mitotic spindle, with its characteristic bipolar shape. Movie 18.5. The two centrosomes that give rise to these microtubules are now called spindle poles, and the interacting microtubules are called interpolar microtubules. Figure 1823. The assembly of the spindle is driven in part by motor proteins associated with the interpolar microtubules that help to cross-link the two sets of microtubules. In the next stage of mitosis, the replicated chromosomes attach to the spindle in such a way that, when the sister chromatids separate, they will be drawn to opposite poles of the cell. Chromosomes attach to the mitotic spindle at prometaphase. Prometaphase starts abruptly with a disassembly of the nuclear envelope, which breaks up into small membrane vesicles. This process is triggered by the phosphorylation and consequent disassembly of nuclear pore proteins and the intermediate filament proteins of the nuclear lamina, the network of fibrous proteins that underlies and stabilizes the nuclear envelope. See Figure 17.7. The spindle microtubules, which have been lying in wait outside the nucleus, now gain access to the replicated chromosomes and capture them. See Panel 18.1, page 626. The spindle microtubules end up attached to the chromosomes through specialized protein complexes called kinetochores, which assemble on the condensed chromosomes during late prophase. Figure 1824. As discussed earlier, each replicated chromosome consists of two sister chromatids joined along their length, and each chromatid is constricted at a region of specialized DNA sequence called the centromere. See Figure 1819b. Just before pro-metaphase, kinetochore proteins assemble into a large complex on each centromere. Each duplicated chromosome therefore has two kinetochores, one on each sister chromatid, which face in opposite directions. Kinetochore assembly depends on the presence of the centromere DNA sequence. In the absence of this sequence, kinetochores fail to assemble, and consequently, the chromosomes fail to segregate properly during mitosis. Once the nuclear envelope has broken down, a randomly probing microtubule encountering a chromosome will bind to it, thereby capturing the chromosome. The microtubule eventually attaches to the kinetochore, and this kinetochore microtubule links the chromosome to a spindle pole. See Figure 1824 and Panel 18.1, page 626. Because kinetochores on sister chromatids face in opposite directions, they tend to attach to microtubules from opposite poles of the spindle, so that each replicated chromosome becomes linked to both spindle poles. The attachment to opposite poles, called bi-orientation, generates tension on the kinetochores, which are being pulled in opposite directions. This tension signals to the sister kinetochores that they are attached correctly and are ready to be separated. The cell cycle control system monitors this tension to ensure correct chromosome attachment, constituting another important cell cycle checkpoint. See Figures 18.3 and 18.13. The number of microtubules attached to each kinetochore varies among species. Each human kinetochore binds 20 to 40 microtubules, for example, whereas a yeast kinetochore binds just one. The three classes of microtubules that form the mitotic spindle are shown in Figure 1825. Chromosomes aid in the assembly of the mitotic spindle. Chromosomes are more than passive passengers in the process of spindle assembly. They can stabilize and organize microtubules into functional mitotic spindles. In cells without centrosomes, including all plant cells and some animal cell types, the chromosomes themselves nucleate microtubule assembly, and motor proteins then move and arrange the microtubules and chromosomes into a bipolar spindle. Even in animal cells that normally have centrosomes, a bipolar spindle can still be formed by these means if the centrosomes are removed. Figure 1826. In cells with centrosomes, the chromosomes, motor proteins, and centrosomes work together to form the mitotic spindle. Chromosomes line up at the spindle equator at metaphase. During prometaphase, the chromosomes now attached to the mitotic spindle begin to move about, as if jerked first this way and then that. Eventually, they align at the equator of the spindle, halfway between two spindle poles, thereby forming the metaphase plate. This defines the beginning of metaphase. Figure 1827. Although the forces that act to bring the chromosomes to the equator are not well understood, both the continual growth and shrinkage of the microtubules and the action of microtubule motor proteins are thought to be involved. A continuous balanced addition and loss of tubulin subunits is also required to maintain the metaphase spindle. When tubulin addition to the ends of microtubules is blocked by the drug colchicine, tubulin loss continues until the spindle disappears. The chromosomes gathered at the equator of the metaphase spindle oscillate back and forth, continually adjusting their positions, indicating that the tug-of-war between the microtubules attached to opposite poles of the spindle continues to operate after the chromosomes are all aligned. If one of the pair of kinetochore attachments is artificially severed with a laser beam during metaphase, the entire chromosome immediately moves towards the pole to which it remains attached. Similarly, if the attachment between sister chromatids is cut, the two daughter chromosomes separate and move toward opposite poles. These experiments show that the replicated chromosomes at the metaphase plate are held there under tension. Evidently, the forces that will ultimately pull the sister chromatids apart again operating as soon as microtubules attach to the kinetochores. Begin operating as soon as microtubules attach to the kinetochores. Proteolysis triggers sister chromatid separation and the completion of mitosis. Anaphase begins abruptly with the release of the cohesin linkage that holds the sister chromatids together. See Figure 1815. This allows each chromatid to be pulled to the spindle pole to which it is attached. Figure 1828. This movement segregates the two identical sets of chromosomes to opposite ends of the spindle. See Panel 18.1, page 627. The cohesin linkage is destroyed by a protease called separase, which up to the beginning of anaphase is held in an inactive state by binding to an inhibitory protein called cyclin. At the beginning of anaphase, cyclin is targeted for destruction by a protein complex called the anaphase promoting complex, APC. Once cyclin has been removed, separase is then free to break the cohesin linkages. Figure 1829. The APC not only triggers the degradation of cohesins, but also targets M-cyclin for destruction, thus rendering the M-CDK complex inactive. This rapid inactivation of M-CDK helps to initiate the exit from mitosis. Chromosomes segregate during anaphase. Once the sister chromatids separate, they are pulled to the spindle pole to which they are attached. They all move at the same speed, which is typically about 1 micrometer per minute. The movement is a consequence of two independent processes that involve different parts of the mitotic spindle. The two processes are called anaphase A and anaphase B, and they occur more or less simultaneously. In anaphase A, the kinetochore microtubules shorten by depolymerization, and the attached chromosomes move poleward. In anaphase B, the spindle poles themselves move apart, further contributing to the segregation of the two sets of chromosomes. Figure 1830. The driving force for the movement of anaphase A is thought to be provided mainly by the microtubule-associated motor proteins operating at the kinetochore, aided by the shortening of kinetochore microtubules. The loss of tubulin subunits from the kinetochore microtubules depends on a motor-like protein that is bound to both the microtubule and the kinetochore and uses the energy of ATP hydrolysis to remove tubulin subunits from the microtubule. In anaphase B, the spindle poles and the two sets of chromosomes move farther apart. The driving forces for this movement are thought to be provided by two sets of motor proteins. Members of the kinesin and dynin families. See Figure 1720, operating on different types of spindle microtubules. One set of motor proteins acts on the long overlapping interpolar microtubules that form the spindle itself. These motor proteins slide the interpolar microtubules from opposite poles past one another at the equator of the spindle, pushing the spindle poles apart. The other set operates on the astral microtubules that extend from the spindle poles and point away from the spindle equator and toward the cell periphery. These motor proteins are thought to be associated with the cell cortex, which underlies the plasma membrane, and they pull each pole toward the adjacent cortex and away from the other pole. See Figure 1830. Unattached chromosomes block sister chromatid separation. If a dividing cell were to begin to segregate its chromosomes before all the chromosomes were properly attached to the spindle, one daughter would receive an incomplete set of chromosomes, while the other daughter would receive a surplus. Both situations could be lethal for the cell. Thus, a dividing cell must ensure that every last chromosome is attached properly to the spindle before it completes mitosis. To monitor chromosome attachment, the cell makes use of a negative signal. Unattached chromosomes send a stop signal to the cell cycle control system. Although the exact nature of the signal remains elusive, we know that it inhibits further progress through mitosis by blocking the activation of the APC. Without active APC, the sister chromatids remain glued together. Thus, none of the duplicated chromosomes can be pulled apart until every chromosome has been positioned correctly on the mitotic spindle. This so-called spindle assembly checkpoint controls exit from mitosis. See Figure 18.3 and 18.13. The nuclear envelope reforms at telophase. By the end of anaphase, the daughter chromosomes have separated into two equal groups, one at each pole of the spindle. During telophase, the final stage of mitosis, the mitotic spindle disassembles, and a nuclear envelope reassembles around each group of chromosomes to form the two daughter nuclei. Vesicles of nuclear membrane first cluster around individual chromosomes and then fuse to reform the nuclear envelope. See Panel 18.1, page 627. During this process, the nuclear pore proteins and nuclear lamins that were phosphorylated during pro-metaphase are now dephosphorylated, which allows them to reassemble and form the nuclear envelope and nuclear lamina respectively. Figure 1831. Once the nuclear envelope has reformed, the pores pump in nuclear proteins, the nucleus expands, and the condensed mitotic chromosomes decondense into their interphase state. As a consequence of decondensation, gene transcription is able to resume. A new nucleus has been created, and mitosis is complete. All that remains is for the cell to complete its division into two separate daughter cells. Cytokinesis. Cytokinesis, the process by which the cytoplasm is cleaved in two, completes M phase. It usually begins in anaphase, but is not completed until the two daughter nuclei have formed in telophase. Whereas mitosis depends on a transient microtubule-based structure, the mitotic spindle, cytokinesis in animal cells depends on a transient structure based on actin and myosin filaments, the contractile ring. See Figure 1820. Both the plane of cleavage and the timing of cytokinesis, however, are determined by the mitotic spindle. The mitotic spindle determines the plane of cytoplasmic cleavage. The first visible sign of cytokinesis in animal cells is a puckering and furrowing of the plasma membrane that occurs during anaphase. Figure 1832. The furrowing invariably occurs in a plane that runs perpendicular to the long axis of the mitotic spindle. This positioning ensures that the cleavage furrow cuts between the two groups of segregated chromosomes so that each daughter cell receives an identical and complete set of chromosomes. If the mitotic spindle is deliberately displaced using a fine glass needle, as soon as the furrow appears, the furrow disappears and a new one develops at a site corresponding to the new spindle location and orientation. Once the furrowing process is well underway, however, cleavage proceeds even if the mitotic spindle is artificially sucked out of the cell or depolymerized using the drug colchicine. How the mitotic spindle dictates the position of the cleavage furrow is still uncertain, but it seems that, during anaphase, both the astral microtubules and the interpolar microtubules and their associated proteins signal to the cell cortex to initiate the assembly of the contractile ring at a position midway between the spindle poles. Because these signals originate in the anaphase spindle, the mechanism also contributes to the timing of cytokinesis in late mitosis. When the mitotic spindle is located centrally in the cell, the usual situation in most dividing cells, the two daughter cells produced will be of equal size. During embryonic development, however, there are some instances in which the dividing cell moves its mitotic spindles to an asymmetrical position, and consequently, the furrow creates two daughter cells that differ in size. In most cases, the daughters also differ in the molecules they inherit, and they usually develop into different cell types. Special mechanisms are required to position the mitotic spindle eccentrically in such asymmetric divisions. The contractile ring of animal cells is made of actin and myosin. The contractile ring is composed mainly of an overlapping array of actin filaments and myosin filaments. Figure 1833. It assembles at anaphase and is attached to membrane-associated proteins on the cytoplasmic phase of the plasma membrane. Once assembled, the contractile ring is capable of exerting a force strong enough to bend a fine glass needle inserted into the cell before cytokinesis. The sliding of the actin filament against the myosin filaments generates the force. See Figure 1739, much as it does during muscle contraction. Unlike the contractile apparatus in muscle, however, the contractile ring is a transient structure. It assembles to carry out cytokinesis, gradually becomes smaller as cytokinesis progresses, and disassembles completely once the cell has been cleaved in two. Cell division in many animal cells is accompanied by large changes in cell shape and a decrease in the adherence of the cell to its neighbors, to the extracellular matrix, or to both. These changes result in part from the reorganization of actin and myosin filaments in the cell cortex, only one aspect of which is the assembly of the contractile ring. Mammalian fibroblasts in culture, for example, spread out flat during interphase, as a result of the strong adhesive contacts they make with the surface they are growing on, called the substratum. As the cells enter M-phase, however, they round up. The cells change shape in part because some of the plasma membrane proteins responsible for attaching the cells to the substratum, the integrins, discussed in Chapter 20, become phosphorylated and thus weaken their grip. Once cytokinesis is complete, the daughter cells re-establish their strong contacts with the substratum and flatten out again. Figure 1834. When the cells divide in an animal tissue, this cycle of attachment and detachment presumably allows the cells to rearrange their contacts with neighboring cells and with the extracellular matrix, so that the new cell produced by cell division can be accommodated within the tissue. Cytokinesis in plant cells involves the formation of a new cell wall. The mechanism of cytokinesis in higher plants is entirely different from that in animal cells, presumably because plant cells are surrounded by a tough cell wall, discussed in Chapter 20. The two daughter cells are separated not by the action of a contractile ring at the cell surface, but instead by the construction of a new wall that forms inside the dividing cell. The positioning of this new wall precisely determines the position of the two daughter cells relative to neighboring cells. Thus, the planes of cell division, together with cell enlargement, determine the final form of the plant. The new cell wall starts to assemble in the cytoplasm between the two sets of segregated chromosomes at the start of telophase. The assembly process is guided by a structure called the phragmoplast, which is formed by the remains of the interpolar microtubules at the equator of the old mitotic spindle. Small membrane-enclosed vesicles, largely derived from the Golgi apparatus and filled with polysaccharides and glycoproteins required for the cell wall matrix, are transported along the microtubules to the phragmoplast. Here, they fuse to form a disc-like membrane-enclosed structure, which expands outward by further vesicle fusion until it reaches the plasma membrane and original cell wall and divides the cell in two. Figure 1835. Later, cellulose microfibrils are laid down within the matrix to complete the construction of the new cell wall. Membrane-enclosed organelles must be distributed to daughter cells when a cell divides. Organelles such as mitochondria and chloroplasts cannot assemble spontaneously from their individual components. They arise only from the growth and division of the pre-existing organelles. Likewise, cells cannot make a new endoplasmic reticulum, ER, or Golgi apparatus unless some part of it is already present, which can then be enlarged. How, then, are these various membrane-enclosed organelles segregated when the cell divides so that each daughter gets them? Organelles such as mitochondria and chloroplasts are usually present in large numbers and will be safely inherited if, on average, their numbers simply double once each cell cycle. The ER in interphase cells is continuous with a nuclear membrane and is organized by the microtubule cytoskeleton. See Figure 1718a. Upon entry into M phase, the reorganization of the microtubules releases the ER. In most cells, the released ER remains intact during mitosis and is cut in two during cytokinesis. The Golgi apparatus fragments during mitosis. The fragments associate with the spindle microtubules via motor proteins, thereby hitching a ride into the daughter cells as the spindle elongates in anaphase. Other components of the cell, including all of the soluble proteins, are inherited randomly when the cell divides. Having discussed how cells divide, we now turn to the general problem of how the size of an animal or an organ is determined, which leads us to consider how cell number and cell size are controlled. Control of cell number and cell size. A fertilized mouse egg and a fertilized human egg are similar in size, and yet an adult mouse is much smaller than an adult human. What are the differences between the control of cell behavior in humans and mice that generate such differences in size? The same fundamental question can be asked about each organ and tissue in an individual's body. What adjustment of cell behavior explains the length of an elephant's trunk or the size of its brain or its liver? These questions are largely unanswered, but it is at least possible to say what the ingredients of an answer must be. Three fundamental processes largely determine organ and body size: cell growth, cell division, and cell death. Each of these processes, in turn, depends on programs intrinsic to the individual cell and is regulated by signals from other cells in the body. In this section, we first consider how organisms eliminate unwanted cells by a form of programmed cell death called apoptosis. We then discuss how extracellular signals stimulate cell survival, cell growth, and cell division, and thereby help to control the size of an animal and its organs. We conclude the section with a brief discussion of the inhibitory extracellular signals that help keep these processes in check. Apoptosis helps regulate animal cell numbers. The cells of a multicellular organism are members of a highly organized community. The number of cells in this community is tightly regulated, not simply by controlling the rate of cell division, but also by controlling the rate of cell death. If cells are no longer needed, they can commit suicide by activating an intracellular death program, a process called programmed cell death. In animals, by far the most common form of programmed cell death is called apoptosis, from a Greek word meaning falling off, as leaves fall from a tree. The amount of apoptosis that occurs in both developing and adult animal tissues can be astonishing. In the developing vertebrate nervous system, for example, more than half of the nerve cells produced normally die soon after they are formed. In a healthy adult human, billions of cells die in the bone marrow and intestine every hour. It seems remarkably wasteful for so many cells to die, especially as the vast majority are perfectly healthy at the time they kill themselves. What purpose does this massive self-suicide serve? In some cases, the answers are clear. Mouse paws and our own hands and feet are sculpted by apoptosis during embryonic development. They start out as spade-like structures, and the individual fingers and toes separate because the cells between them die. Figure 1836. In other cases, cells die when the structure they form is no longer needed. When a tadpole changes into a frog at metamorphosis, the cells in the tail die and the tail, which is not needed in the frog, disappears. Figure 1837. In these cases, the unneeded cells die by apoptosis. In adult tissues, cell death usually exactly balances cell division, unless the tissue is growing or shrinking. If part of the liver is removed in an adult rat, for example, liver cells proliferate to make up the loss. Conversely, if a rat is treated with a drug phenobarbital, which stimulates liver cell division, the liver enlarges. However, when the phenobarbital treatment is stopped, apoptosis in the liver greatly increases until the organ has returned to its original size, usually within a week or so. Thus, the liver is kept at a constant rate through regulation of both the cell death rate and the cell birth rate. Apoptosis is mediated by an intracellular proteolytic cascade. Cells that die as a result of acute injury typically swell and burst, spilling their contents all over their neighbors, a process called cell necrosis. Figure 1838a. This eruption triggers a potentially damaging inflammatory response. By contrast, a cell that undergoes apoptosis dies neatly, without damaging its neighbors. A cell in the throes of apoptosis shrinks and condenses. Figure 1838b. The cytoskeleton collapses, the nuclear envelope disassembles, and the nuclear DNA breaks up into fragments. Movie 18.7. Most importantly, the cell surface is altered in such a manner that it immediately attracts phagocytic cells, usually specialized phagocytic cells called macrophages. See Figure 1532b. These cells engulf the apoptotic cell before it spills its contents. Figure 1838c. This rapid removal of the dying cell avoids the damaging consequences of cell necrosis, and also allows the organic components of the apoptotic cell to be recycled by the cell that ingests it. The machinery that is responsible for apoptosis seems to be similar in all animal cells. It involves the caspase family of proteases, the members of which are made as inactive precursors called procaspases. Procaspases are typically activated by proteolytic cleavage in response to signals that induce apoptosis. The activated caspases cleave and thereby activate other members of the procaspase family, resulting in an amplifying proteolytic cascade. Figure 1839. They also cleave other key proteins in the cell. One of the caspases, for example, cleaves the lamin proteins, which form the nuclear lamina underlying the nuclear envelope. This cleavage causes the irreversible breakdown of the nuclear lamina. See Figure 1831. In this way, the cell dismantles itself quickly and cleanly, and its corpse is rapidly taken up and digested by another cell. Activation of the apoptotic program, like entry into a new stage of the cell cycle, is usually triggered in an all-or-none fashion. The proteolytic cascade is not only destructive and self-amplifying, but also irreversible. Once a cell reaches a critical point along the path to destruction, it cannot turn back. Thus, it is important that the decision to die is tightly controlled. The death program is regulated by the BCL2 family of intracellular proteins. All nucleated animal cells contain the seeds of their own destruction. In these cells, inactive procaspases lie waiting for a signal to destroy the cell. It is therefore not surprising that caspase activity is tightly regulated inside the cell to ensure that the death program is held in check until it is needed. The main proteins that regulate the activation of procaspases are members of the BCL2 family of intracellular proteins. Some members of this protein family promote procaspase activation and cell death, whereas others inhibit these processes. Two of the most important death-promoting family members are proteins called BACs and BAC. These proteins activate procaspase indirectly by inducing the release of cytochrome C from mitochondria into the cytosol. Cytochrome C promotes the assembly of a large, 7-armed pinwheel-like structure that recruits specific procaspase molecules, forming a protein complex called an apoptosome. The procaspase molecules become activated within the apoptosome, triggering a caspase cascade that leads to apoptosis. Figure 1840. BACs and BAC proteins are themselves activated by other death-promoting members of the BCL2 family, which are produced or activated by various insults to the cell, such as DNA damage. Other members of the BCL2 family, including BCL2 itself, act to inhibit, rather than promote, procaspase activation and apoptosis. One way in which they do so is by blocking the ability of BACs and BAC to release cytochrome C from mitochondria. Some of the BCL2 family members that promote apoptosis, including a protein called BAD, do so by binding to and blocking the activity of BCL2 and other death-suppressing members of the BCL2 family. See Figure 1634. The balance between the activities of pro-apoptotic and anti-apoptotic members of the BCL2 family largely determines whether a mammalian cell lives or dies by apoptosis. The intracellular death program is also regulated by signals from other cells, which can either activate or suppress the program. Indeed, cell survival, cell division, and cell growth are all regulated by extracellular signals, which together help multicellular organisms control cell number and cell size, as we now discuss. Animal cells require extracellular signals to survive, grow, and divide. Unicellular organisms such as bacteria and yeasts tend to grow and divide as fast as they can, and their rate of proliferation depends largely on the availability of nutrients in the environment. The cells in a multicellular organism, by contrast, are controlled so that an individual cell survives only when it is needed and divides only when another cell is required, either to allow tissue growth or to replace cell loss. For either tissue growth or cell replacement, cells must grow before they divide. Thus, for an animal cell to survive, grow, or divide, nutrients are not enough. It must also receive chemical signals from other cells, usually its neighbors. Most of the extracellular signal molecules that influence cell survival, cell growth, and cell division are either soluble proteins secreted by other cells or proteins bound to the surface of other cells or the extracellular matrix. Although most act positively to stimulate one or more of these cell processes, some act negatively to inhibit a particular process. The positively acting signal proteins can be classified on the basis of their function into three major categories. 1. Survival factors promote cell survival, largely by suppressing apoptosis. 2. Mitogens stimulate cell division, primarily by overcoming the intracellular braking mechanisms that tend to block progression through the cell cycle. 3. Growth factors stimulate cell growth, an increase in cell size and mass by promoting the synthesis and inhibiting the degradation of proteins and other macromolecules. These categories are not mutually exclusive, as many signal molecules have more than one of these functions. Unfortunately, the term growth factor is often used as a catch-all phrase to describe a protein with any of these functions. Indeed, the phrase cell growth is often used incorrectly to mean an increase in cell number, which is more correctly termed cell proliferation. In the following sections, we examine each of these types of signal molecules in turn. Animal cells require survival factors to avoid apoptosis. Animal cells need signals from other cells to survive. If deprived of such survival factors, cells activate their intracellular suicide program and die by apoptosis. This requirement for signals from other cells helps to ensure that cells survive only when and where they are needed. Nerve cells, for example, are produced in excess in the developing nervous system and then compete for limited amounts of survival factors that are secreted by the target cells they contact. Nerve cells that receive enough survival factor live, while the others die by apoptosis. In this way, the number of surviving nerve cells is automatically adjusted so that it is appropriate for the number of cells with which they connect. Figure 1841. A similar dependence on survival signals from neighboring cells is thought to control cell numbers in other tissues, both during development and in adulthood. Survival factors usually act by binding to cell surface receptors. These activated receptors then turn on intracellular signaling pathways that keep the death program suppressed, usually by regulating members of the BCL2 family of proteins. Some survival factors, for example, increase the production of BCL2, a protein that suppresses apoptosis. Figure 1842. Mitogens stimulate cell division. Most mitogens are secreted signal proteins that bind to cell surface receptors. When activated by mitogen binding, these receptors initiate various intracellular signaling pathways, discussed in Chapter 16, that stimulate cell division. These signaling pathways act mainly by releasing the molecular brakes that block the transition from the G1 phase of the cell cycle into S phase. An important example of such a molecular brake is the retinoblastoma RB protein, first identified through studies of a rare childhood eye tumor called retinoblastoma, in which the RB protein is missing or defective. The RB protein is abundant in the nucleus of all vertebrate cells. It binds to particular transcription regulators, preventing them from stimulating the transcription of genes required for cell proliferation. Mitogens release the RB brake in the following way. They activate intracellular signaling pathways that lead to the activation of the G1 CDK and G1S CDK complexes discussed earlier. These complexes phosphorylate the RB protein, altering its conformation so that it releases its bound transcription regulators, which are then free to activate the genes required for cell proliferation. Figure 1843. Most mitogens have been identified and characterized by their effects on cells in culture. Figure 1844. One of the first mitogens identified in this way was platelet-derived growth factor, or PDGF, the effects of which are typical of many other discovered since. When blood clots form in a wound, for example, blood platelets incorporated in the clots are triggered to release PDGF. PDGF then binds to receptor tyrosine kinases, discussed in Chapter 16, in surviving cells at the wound site, stimulating them to proliferate and help heal the wound. Similarly, if part of the liver is lost through surgery or acute injury, cells in the liver and elsewhere produce a protein called hepatocyte growth factor, which helps stimulate the surviving liver cells to proliferate. Growth factors stimulate cells to grow. The growth of an organism or organ depends on cell growth as much as on cell division. If cells divided without growing, they would get progressively smaller, and there would be no increase in total cell mass. In single-celled organisms such as yeast, cell growth, like cell division, requires only nutrients. In animals, by contrast, both cell growth and cell division depend on signals from other cells. Cell growth, however, unlike cell division, does not depend on the cell cycle control system, in either yeast or animal cells. Indeed, many animal cells, including nerve cells and most muscle cells, do most of their growing after they have become specialized and permanently stop dividing. Like most survival factors in mitogens, most extracellular growth factors bind to cell surface receptors, which activate various intracellular signaling pathways. These pathways lead to the accumulation of proteins and other macromolecules, and they do so by both increasing the rate of synthesis of these molecules and decreasing the rate of degradation, figure 1845. Some extracellular signal proteins, including PDGF, can act as both growth factors and mitogens, stimulating both cell growth and progression through the cell cycle. Such proteins help ensure that cells maintain their appropriate size as they proliferate. Compared to cell division, there has been surprisingly little study of how cell size is controlled in animals. As a result, it remains a mystery how different cell types in the same animal grow to be so different in size. Figure 1846. Some extracellular signal proteins inhibit cell survival, division, or growth. The extracellular signal proteins that we have discussed so far, survival factors, mitogens, and growth factors, act positively to increase the size of organs and organisms. Some extracellular signal proteins, however, act to oppose these positive regulators and thereby inhibit tissue growth. Myostatin, for example, is a secreted signal protein that normally inhibits the growth and proliferation of the myoblasts that fuse to form skeletal muscle cells during mammalian development. When the gene that encodes myostatin is deleted in mice, their muscles grow to be several times larger than normal, because both the number and the size of muscle cells is increased. Remarkably, 2 breeds of cattle that were bred for large muscles turned out to have mutations in the gene encoding myostatin. Figure 1847. As we discuss in the final chapter, cancers are similarly the products of mutations that set cells free from the normal social controls operating on cell survival, growth, and proliferation. Because cancer cells are generally less dependent than normal cells on signals from other cells, they can outsurvive, outgrow, and outdivide their normal neighbors, producing tumors that can kill their host. In this chapter, when we have discussed cell division, we have always been referring to those ordinary divisions that produce 2 daughter cells, each with a full and identical complement of the parent cell's genetic material. There is, however, a different and highly specialized type of cell division called meiosis, which is required for sexual reproduction in eukaryotes. In the next chapter, we describe the special features of meiosis and how they underlie the genetic principles that define the laws of inheritance. Essential concepts. The eukaryotic cell cycle consists of several distinct phases. These include S phase, during which the nuclear DNA is replicated, and M phase, during which the nucleus divides, mitosis, and then the cytoplasm divides, cytokinesis. In most cells, there is one gap phase, G1, after M phase and before S phase, and another G2 after S phase and before M phase. These gaps give the cell more time to grow and to prepare for the events of S phase and M phase. The cell cycle control system coordinates the events of the cell cycle by sequentially and cyclically switching on the appropriate parts of the cell cycle machinery and then switching them off. The control system depends on a set of protein kinases, each composed of a regulatory subunit called a cyclin and a catalytic subunit called a cyclin-dependent protein kinase, CDK. Cyclin concentrations rise and fall at specific times in the cell cycle, helping to trigger events of the cycle. The CDKs are cyclically activated by both cyclin binding and the phosphorylation of some amino acids and the dephosphorylation of others. When activated, CDKs phosphorylate key proteins in the cell. Different cyclin CDK complexes trigger different steps of the cell cycle. mCDK drives the cell into mitosis. G1-CDK drives it through G1. G1-SCDK and SCDK drive it into S phase. The control system also uses protein complexes that trigger the proteolysis of specific cell cycle regulators at particular stages of the cycle. The cell cycle control system can halt the cycle at specific checkpoints to ensure that intracellular and extracellular conditions are favorable and that the next step in the cycle does not begin before the previous one has finished. Some of these checkpoints rely on CDK inhibitors that block the activity of one or more cyclin CDK complexes. SCDK initiates DNA replication during S phase and helps ensure that the genome is copied only once. Checkpoints in G1, S phase, and G2 prevent cells from replicating damaged DNA. mCDK drives the cell into mitosis with the assembly of the microtubule-based mitotic spindle, which will move daughter chromosomes to opposite poles of the cell. Microtubules grow out from the duplicated centrosomes, and some interact with microtubules growing from the opposite pole. thereby becoming the interpolar microtubules that form the spindle. Centrosomes, microtubule-associated motor proteins, and the replicated chromosomes themselves work together to assemble the spindle. When the nuclear envelope breaks down, the spindle microtubules invade the nuclear area and capture the replicated chromosomes. The microtubules bind to protein complexes, called kinetochores, associated with the centromere of each sister chromatid. Microtubules from opposite poles pull in opposite directions on each replicated chromosome, bringing the chromosomes to the equator of the mitotic spindle. The sudden separation of sister chromatids allows the resulting daughter chromosomes to be pulled to opposite poles by the spindle. The two poles also move apart, further separating the two sets of chromosomes. The movement of chromosomes by the spindle is driven both by microtubule motor proteins and by microtubule polymerization and depolymerization. A nuclear envelope reforms around the two sets of segregated chromosomes to form two new nuclei, thereby completing mitosis. The Golgi apparatus breaks into many smaller fragments during M phase, ensuring an even distribution between the daughter cells. In animal cells, cytoplasmic division is mediated by a contractile ring of actin filaments and myosin filaments, which assembles midway between the spindle poles and contracts to divide the cytoplasm in two. In plant cells, by contrast, cell division occurs by the formation of a new cell wall inside the parent cell, which divides the cytoplasm in two. Animal cell numbers are regulated by a combination of intracellular programs and extracellular signals that control cell survival, cell growth, and cell proliferation. Many normal cells die by apoptosis during the lifetime of an animal. They do so by activating an internal suicide program and killing themselves. Apoptosis depends on a family of proteolytic enzymes called caspases, which are made as inactive precursors, procaspases. The procaspases are themselves often activated by proteolytic cleavage mediated by caspases. Most animal cells require continuous signaling from other cells to avoid apoptosis. This may be a mechanism to ensure that cells survive only when and where they are needed. Animal cells proliferate only if stimulated by extracellular mitogens produced by other cells, ensuring that a cell divides only when another cell is needed. The mitogens activate intracellular signaling pathways to override the normal brakes that otherwise block cell cycle progression. For an organism or an organ to grow, cells must grow as well as divide. Animal cell growth depends on extracellular growth factors, which stimulate protein synthesis and inhibit protein degradation. Cell and tissue size can also be influenced by inhibitory extracellular signal proteins that oppose the positive regulators of cell survival, cell growth, and cell division. Cancer cells fail to obey these normal social controls on cell behavior and therefore outgrow, outdivide, and outsurvive their normal neighbors.